Welcome to our immunity whiteboard animation.
We use the castle metaphor to model the immune system. Mast cells, shown as the scout, release histamine, which then cause leaky capillaries and dilated arteries. The capillaries and arteries are the tunnels and secret paths in the castle, with the histamine as the road builders and carriers of the messages.
The effects of histamine facilitate easier access for phagocytosis by neutrophils and macrophages, and once bacteria is gone, it allows for healing of tissue.
A tower of the castle would be the antigen-presenting cell, which engulfs a pathogen and degrades it, and displays antigen fragments with class II MHC molecules on the cell's surface.
Similar to a castle's messenger birds, helper T cells detect the message and deliver it. The mechanism that occurs involves helper T cells which bind to the APC's MHC class II complex with the help of CD4 accessory proteins. Cytokines are then released to activate other helper T cells or the humoral and cell-mediated response. So going into more detail about cell-mediated immunity, helper T cells act as a messenger bird that deliver the cytokine messages to activate cytotoxic T cells.
This gives rise to memory cytotoxic T cells, which are the historians in our analogy, and active cytotoxic T cells, which are the army. Active cytotoxic T cells defend against infected cells, cancer, and transplanted tissue.
The activated cytotoxic T cells bind to a class I MHC antigen fragment complex on an infected cell via CD8.
The T cell releases perforin molecules, which form pores in the infected cell membrane, and granzymes, which are enzymes that break down proteins and enter the cell by endocytosis. These enzymes initiate apoptosis within the infected cell, leading to fragmentation of the nucleus and cytoplasm, and eventual cell death.
Humoral immunity occurs in your bloodstream. In this response, B cells are like soldiers, each with specific specialties.
From the bone marrow, lymphocytes turn into B cells or T cells depending on where they mature. Then they continue this training or maturation in the blood, lymph, or spleen.
More specifically, clonal selection begins when an antigen is presented to many lymphocytes in the lymph nodes until a match is made. The selected B cell proliferates and forms clones of itself. Following cloning, B cells are stimulated by helper T cells when they bind via class II MHC and CD4 proteins. B cells can differentiate into plasma cells, similar to castle defenders, that can secrete antibodies, or they can also become memory B cells, which are similar to castle historians. So once a plasma cell releases antibodies, there are a couple of mechanisms in antigen disposal by antibody, such as agglutination. The antibody is almost like a net, clumping bacteria or foreign pathogens in order to promote phagocytosis. In neutralization, an antibody is sent out to bind to antigens on pathogen surfaces to prevent it from entering a host cell. Opsonization is another method that involves the antibodies binding to antigens, serving as markers to promote phagocytosis. The complement system releases complement proteins to form a membrane attack complex, similar to a cannon, which forms pores in the pathogen leading to lysis and cell death. That is our brief overview on immunity, and thank you for watching.